So it's my great pleasure to introduce Jon Sporring, who hopefully will show us, has been downloading some of this data, how we can actually quantify it and make it useful, and many of the techniques have been working. Please, Jon.
Thank you. No, I'm sorry, I will disappoint you. I didn't bring any examples on the HOAHub data, but I will. So what I will show you is some of the things that I've been working on statistically summarizing shapes, because I think this is about where we are. So as you all know, data science and deep learning has taken giant leaps these last few years. And this is an example of Segment Anything, which is software made available on the internet by Meta. You can all go and try it. It is an example of a deep learning model, which has been trained on basically images on the internet. They download them all and train a model. So it's really good at it. And it doesn't require much human interaction. And I've tried it on different microscope images over here without any sort of other input than the image. And this is just what it comes out. The top one is a light microscopy image, and this one is an electron microscopy. That's all my colleagues made. And it does it quite well. In this, there's some sort of automatic scale selection because what's a segment? A segment is, well, there might be parts of it that new things in segment, other parts that I don't think are segment. But in this model, you can even interact with the results in a sort of multi-scale fashion. Now, if you go and try it, you will discover that it works okay on your data. Okay, but possibly not perfect. And this is because when you should think of these models, these foundational models, as some call them, as a car. I think now the car has been built, and we still need to learn to drive it. So there's no shortcut. I think if you come with new data that these models haven't seen, you have to provide lots and lots of examples, and you have to train them. And some of these models are very big. It can take days at least to train them. And then you have to check the result, which I think is another real challenge, and particularly with the HOAHub data of those size. It's a real challenge to figure out whether these models have run something well to your quality standard. So whatever you think about this, don't forget that working with the data afterwards takes enormous amount of time. Okay, now what I've been for many years now working on what we can do after these segmentation models got good enough for us to actually look at not the pixels, not the boxes, but the shapes. And I'll just show you three different examples of ways we've been working with this. The first one is looking at objects in images as points. Secondly, we've extended this to shapes of different kinds. And the last thing that is my favorite topic these days is also to look at sort of topological features of data. Since we started some years ago, and since I'm a computer scientist, for me, these are all just raw pixels. We've been looking very much at the electron microscope images. And this is also due to that well, I have to find collaborators that can pose interesting questions in which these analysis says that I do have some sort of relevance. I want to make programs that are useful for something, not just theoretical investigation. So this is an example of an electron microscope images. We basically worked on the focused ion beam scanning electron microscope. This we have not made some years ago. And we've also looked a bit on zero section emission electron microscope images. And one of the key features of these images is that they have a high resolution, and let's say the landscapes of objects in these images are still not really well understood. And there are lots of them, and this is something that really interests me because suddenly I have a source of interesting objects that have varying shapes and they are spread out in a non-trivial ways. So Statistical summaries of these shapes and their relation is what interests me. So let's start at points. There's in statistics a really well developed model for the study of spatial point processes. And one way to study this, so this could, for example, be the vesicles that you see up here. So these are vesicles, and this is a synapse, and we can recognize the synapse because it's still a bit dark on the other side. So this is an asymmetric synapse. So if we were this particular study was about depression and whether we could see anatomical changes in the distribution of the vesicle depending on whether these rodents that we looked at had been exposed to stress or not. That makes sense because these vesicles are used in the communication between the cells. So if you're stressed, you're ready to communicate. Is the sense. So the basic idea, the tool here that we're using is the Ripley's K function. It's a distance function. So imagine this is the center of the vesicle, and then for a given radius, you count how many other vesicles you see, right? And then you do it for all the other vesicles, and you take the average curve. That's what this K function is. And people typically normalize it just that because that curve, if everything is random, will grow like a radius square, and then you take the square root as a constant, so you get the line in the uniform distribution. And this example that's running down here is this is a hypothetical one I made synthetically, but imagine that this is your lawn at home or my lawn at home, and I have a couple of apple trees, and then I know where did the apples fall. Before near the tree. So that could be a distribution of points. And over here, I have like a null hypothesis that I generated, I statistically can draw from. So I'm regenerating or drawing from it 10 times. And each time for all of these, I measure my KOL function. And what you see over here is this is my yard, the apples fell on my lawn, and this is the null hypothesis. And you can clearly see these curves are very different. And since this is radius, what you can see is that near the tree, there is an overcount of apples, and then it sort of aligns. We think of this as clustering, so this can be used to, for example, in sets like this without imposing too much geometric constraints to figure out whether there's clustering effect of vesicles in this case. And one of the hypotheses we were going for was, is there like a reserve pool of vesicles somewhere ready to be used? Yeah, and Magda over here was a PhD student at this time. She's now in Norway. And she worked on this, so she did a lot of analysis. And one of the things we did with this data was we calculated these curves, so the L functions you saw before. And this is for a single synapse. There were lots of vesicles in this one. And one of the things you can observe with this method is that they have a distance with the radius that's much bigger than expected. So somehow there's something we don't see in these electron microscope images. These vesicles seem to sort of repel each other a little bit, and afterwards there's a bit of an overcount. And when you the green and the blue curve here are null hypothesis that they were just random. So we took these weird shape of cell bodies or cell areas and randomly put points. And so it's not random. That's what we can see. Let's say we can give it sort of interpretation, and I think this is a good tool to go back and then do further investigations. What can be the cause? This is for a single synapse area, but of course you can average across many. I'm showing you images in 2D, but that's just because it's difficult to show images in 3D. But we are really only working on 2D. So one of the problems with this approach is that many of the objects are not well described like points. Okay? So they have weird shapes. The mitochondria, they're really very sausage-like. They're long, and maybe they even have bifurcations, etc. So the next thing was to look at how can we, with simple means, extend this methodology to shapes. And this is what Hans worked on for a while. And the basic idea is that where we before had a distance function that was point-like, we can now think of a distance as a red line. So each of these red lines have the same shortest distance into the moon. And then we can think about how are this distance field, how does this, for example, interact with the star fields? Okay. And with this, you can make similar curves. So now it's not point to point, but it's shape to shapes. And for the stars, you get something like this. In the beginning, near the moon, there's nothing. Then there comes a lump of stars, and there's nothing, and then there's another lump of stars. And we like various versions of it. The derivative of this one, which is well, for if this had been the Ripley's K function as we saw earlier, then the derivative of that would be the pair correlation function, if someone knows this. It's not quite this for this case, but the one we favor is the one where we take the ratio of the count of the how's the area overlap of this extended moon with the stars and divided by the shape of the extended moon. So that's for this case, it goes something like this. And for this case, this gives well, first of all, for the vesicles are round, so well, that works well with points, but this point anyway. And there, from this, we can clearly see that this fractional curve, that there's a build-up of vesicles in the synapse, which is not a surprise, but we could also look at other things like where do we typically observe the mitochondria in relation to the active zone here. And I don't know if it comes as a surprise to you, but it clearly shows, and this is across hundreds of active zones, that the mitochondria is never near the actives. So the blue shaded area here is, of course, a measure of how much variation there is about the sun. So the last thing I will talk about is how one of the exciting things I think is going on these days is the interplay with between geometry and topology. So topology is basically everything you can say without having to mention distances. Okay, so a typical cup has a hole, typical glass does not. So they are quite different. The cup has the same topology as a donut. A simple glass has the same topology as a sphere. And that's a little bit, it takes some twisting of your mind in order to understand how you can use that for anything sensible. And particularly, I think in terms of the pure mathematics of it, it's not so useful for us because a hole could be of any size, but I think size matters in our data. But the last 20 years, there's been a lot of work on what's called topological data analysis, and the basic idea is that you intermix geometry and topology. So, and for this, I love this. I'm proud of this movie. This is an example of crystal membrane that Ken Howell has segmented. So inside these mitochondria, you can see there are crystal membranes, and this has an intricate structure, and these apparently mitochondria are broken down into sub pieces, moved around in the cell, and put together again. And this must have influence on how these crystal membranes are shaped. Anyway, so this is a good case for us to start the topology because, well, what is the structure? There are these tubular-like things. Somewhere this is a little bit more flat, and they are all connected, or maybe they are not. and how does that actually relate to the function. This particular project is about oxygen efficiency. So they have, the biologists I'm working with have a model of mice that do not uptake oxygen properly, and perhaps it's got something to do with the crystal membrane. We're still looking at that. So topological data analysis is what I talk about. And the basic idea is that if you have a point set like this, then you replace each point with a disk or a sphere of the same radius. So that could be one case here, and if they overlap, you draw a connection. Okay, and if there are three that mutually overlap, you replace them by triangle. This is a mix between geometry and topology, because geometry are these spheres, they represent distances, while topology is this weird looking graph. And in this case, we are interested in, for example, the number of holes that appear. So this is not a hole because they are mutually overlapping, but this is an example of a hole of a cycle. And one part of this is counting the number of cycles. So here there are three, here there's one. And what we can use that for is we can see to what extent these cycles are stable or persistent. This is called persistent homology. And the count of these cycles, that's also known as a Betti number. So that for each dimension, you have different ways cycles can appear, and these Betti numbers are just like counting the number of holes in the cup. Just one. And when you then vary R, and this is then the interplay between geometry and topology. When you vary R, what you get is something like this with this shape. At some point, there are lots of holes, and then later, all the holes disappear. Technically, the way we count them is we make something called a barcode, and the red ones up here are the number of connected components. So we start by, what is it, 14, and then when these are joined there, then 13. And the blue ones are the number of cycles, one-dimensional cycles, technically. And at every radius up here, you can just count the number of red bars. That would give you your Betti zero number, and here you would get Betti one or the number of cycles. So that's a relatively simple structure, not necessarily easy to do. And what we've done we've done a slightly different algorithm for this. We've come up with a method to statistically summarize the width of these tubular structures of crystal membranes. So I call them tubes in the sense that if you have a shape, think of it as a circle, and then you just extend it along the top. But it could also be a more flat structure, and you extend it along. And what this thing we've come up with measures the shortest distance here. So we think this is relevant for this case, and it works like this. If you think of this as the size of a crystal membrane, and this is the inside, if I expand this basically by something called dilation, mathematical morphology dilation, then after certain expansions, I've moved my boundary here, and if I move further, these two will overlap, but they will appear. So there's a hole here, and now there is a smaller hole. And if we do this just like before and count the number of holes, we get count curves that have shapes that look like this. This is very schematically, but it's the same idea. And we can figure out what is the actual point, and then we can do statistics on this across a large number of crystal membrane. And so we see that the half, so the radius of these distances is approximately 20. So I think my time is up, but I just wanted to give you a quick peek into what I think is the most important thing to look at from right, where we are. And I gave you 3 simple examples of how we work with method development. And well, if you want to talk about data science and image analysis, you're more than welcome. We have several structures set up for it. There's the image analysis part of the HOAHub, which is well, we have a couple of people there talking. There's also something called the nearby imaging, which we collaborate much with, which has image processing as a service. So I think there's lots of opportunities to get them. Thank you.